and thank you for the invitation to come here today. I'm really excited to have the chance to talk to you all. And I'm going to take a different tack than I usually do and talk about different things. I'm going to try to keep the discussion of the things I usually talk about, like finding spatiotemporal patterns in neural activity and linking those to neurophysiology, kind of to a minimum, because I want to bring up more of a question that I hope will spark some discussion. And I think it's well suited for this meeting. And that's about how do we talk about data that crosses scales? How do we go from activity at one scale to activity at another scale? And I know there's a lot of interest in this because I've heard a lot of other talks kind of mention this in passing. So I'm hoping we can go a little bit deeper into that today. So that said, because this is all very new, I'm going to be talking about these cross-scalar interactions in a brand new set of data that we just got. And some of the analysis I'm going to show you today should all be considered very preliminary. None of it's published. Some of it is barely done. You're going to see by the time we get to the end, I've got data from one mouse that I managed to analyze so far. But I want us to think of this more as a demonstration of what we might do and spark some discussion of other ways that might be better to go about this. So stay tuned. So we all know that there's activity in the brain at many different scales. You can think of something happening at a single synapse, at the millisecond time scale, all throughout the brain. But then there's all of the structure that is imposed upon that synapse level activity. Let's see if I can fix it. There we go. Okay, so there are laminar probabilities of where things interact. I keep hitting this, sorry. I need to not touch it. There are columnar structures that maybe contain collections of neurons that are dedicated to a similar task. There are well-described circuits from things like the thalamus to the cortex that people have gone in and examined in exhaustive detail. We know that there are areas with really widespread projections that can regulate excitability throughout the whole brain, things like the locus coeruleus or the basal nucleus of Meinert in the forebrain. And then on top of all of these neural structures, there's a lot of connectivity in the glial cells that may be important for understanding how activity propagates when we're looking at dynamics throughout the whole brain. I'm especially interested in astrocytes, which have very different connectivity patterns than neurons, but yet may contribute to some of what we see with large-scale imaging modalities. So all of this is going on. And somehow, if we want to understand how things happening at one scale relate to things happening at another scale, we've got to disentangle all of this. So the large scale activity is basically created by this fast local activity, but at the same time, those large scale patterns end up creating these macro scale phenomena like the traveling waves that people have seen that may then constrain or facilitate or modulate that local activity in turn. So we've got this cyclical interaction between scales that I think is really fascinating and really important to understand. So I want to talk just about kind of generalities first. So let's say we've got some way to get this multimodal data. We can get really high spatial and temporal resolution with one modality, low spatial and temporal resolution with another modality. For now, let's just assume we're looking at the same area. That's usually not true. The same sort of sensitivity to the same process, also usually not true. But let's make this as simple as possible. So basically, I have something where I've imaged the same square with 20 millisecond sampling on one side. And let's see, what did I decide? I called that, anyway, 10 times the spatial resolution of what I've got on the other side. So a factor of 10 in space, a factor of 50 in time, difference between these two modalities. And somehow I want to know how what's happening here is related to what's happening over there. And so I think there are some ways that people have addressed this question. And I think they all have good points and bad points. But to start with something that's pretty simple, we could say, okay, well, for now, let's just look at the spatial structure. You guys have seen many graphs at this point I can take my native high spatial resolution data and I can make some kind of graph structure. I can take my lower resolution data and make some kind of graph structure. And maybe I can even compare across graphs somehow. Maybe I can say, oh, I'm going to look at modularity or small worldness or efficiency and see if I see the same kind of things at different scales. So that's really cool. Maybe it's even a little bit too hard. Maybe I say, gosh, I don't know, trying to figure out how to connect this 10,000 mode graph to a 1,000 mode graph. I don't even know how to do that. So let's make it even easier. So then let's say, okay, I'm going to now not just ignore all the timing information or kind of collapse it into one statistic. I'm also going to now downsample in space. And so I could take that really high spatial resolution data and just do some kind of averaging until it looks just like my low resolution data. Or I could even say, well, I don't really need this level, or I'm going to start with a simpler spatial representation. And so I can take both of those sets of data and register them to some kind of atlas, parcellate them up. And now instead of, let's see, this was 40,000 nodes or 400 nodes, now I've got 6. So I can do this kind of simplification. Of course, and that's valuable, that's important, but we know that these hide things that we want to see. We know that, for example, when you're looking at spatial information, there's a lot of symmetry, a lot of redundancy in what happens in the brain. If you give the exact same stimulus to an anesthetized rat, you'll get different patterns of neural activation every time, but the overall response looks the same. So there's this built-in redundancy, and we don't see that when we go to these average scales. There are things like subpopulations. I think a prime example of this is ocular dominance columns, right? You've got these mixed columns, some look at activation in the left eye, some in the right eye, and they're all kind of interwoven with each other. So at small spatial scales, you might get a very structured pattern, but when you go to the coarser spatial scales, they average out to nothing. So we can take this approach, and I'm going to take this approach, but I want to point out that we're missing a lot with this. And so I think this is a gap that I would love to have a better way to solve. Okay, but getting back to time, I mentioned before that my lab has done a lot of work looking at how brain activity evolves, trying to pull out spatiotemporal patterns. So time is really interesting to me. And so we also have to deal, even let's say we figured out the spatial correspondence, we have to deal somehow with the temporal correspondence. So we could take the same approach. We could say, well, in this modality, I've got 10 images per second, and this one I only have 1. I'm just going to average those 10 together and compare it to that 1. Okay, I can do that. You can say, well, maybe I don't actually need the highest frequencies. Maybe I only want to look at the low frequency part. I can filter. I can do some kind of downsampling. All of those are going to throw away that lovely timing information. And since my lab has really been interested in timing, this is the part that we've really tried to focus on first, and we're hoping to come back to stuff about how to maintain the spatial representation maybe later. So a couple of approaches to doing this, and I don't think any of these are ideal. I guess maybe now is a good time to tell you that the internal model in my brain for going across scales is that we have an inherently low representation trajectories for activity at any given scale, right? If I'm looking at cells, if I'm looking at areas, if I'm looking at the whole brain. And so what I would really like to do is relate these low dimensional brain trajectories to each other across spatial and temporal scales. But as a step towards doing that, we've started thinking about things like brain states. So we heard last night about recurrent states that show up, how the brain revisits certain states. We've heard that over and over today. And I think that maybe defining these as brain states gives us one way to kind of start crossing this temporal scale. So the idea is that you have some sort of pattern, some activity pattern that recurs. Maybe not every second, maybe not every image, maybe every 100 or every 50, but something that keeps coming up. And so maybe we can describe the brain's activity in terms of these states. And I'm going to come back to ways to do this. This is just kind of a generic description right now. So maybe I can make most of my activity patterns out of these 4 states at the faster scale, out of those 4 states at the slower scale. And then the nice thing is, this maybe I could go back and keep that original temporal resolution. So I can say patterns at the fast scale happen at these times, and I can say, okay, this is pattern 1, 2, 1, 2, 3, 4, 1, and go through that time course. I can do the same thing for the slower scale in a time walk fashion. And then I can start to ask questions about how states at one scale correspond to states at another scale. So that's one approach, and we're playing with that just a little bit. Another thing that you could imagine doing is saying, well, the large scale, slower activity maybe modulates those faster patterns of activity. So maybe I want to do something where I look at, say, the phase of a slow pattern, a slow trajectory of activity, and then the amplitude or the incidence of a particular state of that faster activity. And so phase amplitude coupling, I think, is the classic example of this, where people will do things like electrophysiology and say, oh, you have some sort of slower carrier wave that modulates the amplitude of your higher frequency. So I think that's another approach that maybe we can use. Okay, so that was my overview of this problem, this challenge, this gap that I think that I would like to hear everybody discuss. And I'm going to try to come back now to what we've actually done and what our data looks like to give you guys some kind of concrete examples of how this might work. So I mentioned that my lab is interested in the dynamics of whole brain activity, this low dimensional representation. And so we want to identify some kind of parsimonious representations of what's going on in your brain. We use resting state fMRI. It's not directly sensitive to neural activity, which is a challenge, but it has really a nice combination of spatial and temporal resolution and whole brain coverage. And it's non-invasive, which means we can use it in just about any species or any situation that we want to use it in. So we have spent a lot of time trying to link these parsimonious descriptions to some sort of neurophysiological underpinnings. And I'm barely going to talk about that today, but we've been doing all of this to try to get to some kind of linking mechanistically of what's going on across scales. Okay, so we're really interested in the intrinsic activity of the brain. We've talked a lot about tasks so far, and a task, of course, is fascinating, but we want to understand kind of this intrinsic brain activity first. If you look at the importance, of what's going on, we know brain activity is important because it consumes a lot of the body's energy. And if you look at how that's distributed, the amount that goes to the task is fairly small compared to what's going on just to keep your brain, doing what it does. We know that this background activity is influential. If you give a different stimulus, you can get different patterns of response depending on what's going on in that intrinsic activity at that time. And we know that this is really persistent. And I think this is a really interesting study. They were actually looking at a particular network during wakefulness, during deep sleep, and showing that there are a few differences. But what really strikes me is how similar these patterns are, right? That structure is still there. We can see similar structure across species, across conditions, from anesthetized rats all the way up to awake humans. So there's something really fundamental going on that I think it's important to understand if we want to understand how the brain functions. Now, if you've done resting state fMRI, there are many, many ways to analyze this data, and they proliferate every year. So I'm sorry if I don't have your favorite one up on the screen. We're going to use just a couple that I think give specific types of insight into what's going on. We're going to talk about correlation matrices, which are the same things that describe that overall spatial structure, right? The idea that, well, let's ignore time, let's just look at spatial structure of the coordination, I should say. Coactivation patterns, which is more or less this definition of a brain state at a time by time view. So short-lived transient patterns of activity that repeat over time. And then we're going to look at quasi-periodic patterns, which, if coactivation patterns are snapshots, quasi-periodic patterns are movies. So describing the brain activity in terms of these spatiotemporal trajectories that occur repeatedly and contain a specific sequence of activation across areas. So these capture time-lagged relationships in addition to the time-locked relationships that we usually think about in terms of things like correlation or coactivation patterns. Okay, so I suspect you guys all know what functional connectivity matrices look like. We have either voxel by voxel or parcel by parcel matrix where we calculate the correlation pairwise between everything. And then we can organize this into modules if we want to understand something about the structure of the brain. And it doesn't matter how much time we use for this, right? You may get a more or less accurate representation of the actual structure of the correlation data. But you can theoretically do this on a 10-second window or a 10,000-second window, and it'll look the same way. It's collapsing the temporal dimension. So that's handy when we're going across scales because we don't have to worry about that timing part. So I mentioned the coactivation patterns. To be a little bit more specific about what these are in terms of resting state fMRI data, people have for years known that if you look at correlation between two areas, it's really driven by a few high amplitude events. And so for coactivation patterns, they said, well, let's not look at all of this stuff because maybe some of that's pretty noisy, right? Let's just take those high amplitude events and see if we can use those to identify the spatial structure of coordination in the data. And what they showed is if you do that, and you take just a few high amplitude events from every run and start averaging the ones that are similar together, you account for most of the correlation structure. So, this is one of the things that drives what we see in, say, a correlation matrix, but it lets us have that temporal information. The quasi-periodic patterns you can think of as being almost the same thing, but instead of pulling out high amplitude instances of a single pattern of activity, we're getting a movie of activity. And so we're finding those, we do some averaging, pull out typical patterns. There's a couple of different algorithms you can use, but they make movies. And let's see if I can make this play. That's a good question. I'm gonna do it on here. Okay, so this is what one of the movies looks like. And they cover about 20 seconds for awake humans. And these are truly whole brain patterns. So if you zoom in on the subcortex and the cerebellum, you can see that they kind of follow that same timing and they activate with certain areas of the cortex as well. So these patterns really describe activity throughout the whole brain. Oh, and now I'm going backwards. There we go. Okay, so one of the things I like about this is that they are suited for that type of analysis we talked about. If you want to look at, say, phase amplitude relationships, there's a few algorithms. We use QPP or quasi-complex PCA to usually pull out these things. And just like the coactivation patterns, if you take those out, which has been done in this half of the functional connectivity matrix, you account for most of the structure that was there before. So just like the high amplitude instances of activity, these movies are really describing most of what's going on in the brain. Okay, we use in general about 3 patterns, maybe 4 for the quasi-periodic patterns based on how fast the variance drops off. Coactivation patterns, there's not really a set number. People use anything from like 4 to like 20 or 30, depending on how much data they have. And so I think that's still kind of something that we need to figure out what's the right number. Okay, so I told you I'm not going to go too deep into all of the stuff that we've been looking into about, where do these patterns come from? So I want to just give you some hints that they're interesting. We know they're not related to cognition or information processing. They're still there when you're anesthetized. We've done a lot of work to show that they're not related just to motion or respiration. That's out there. So there are some ways that neural activity could give rise to these types of whole brain patterns. One idea is that those small nuclei that I mentioned with the far-ranging inputs might be able to coordinate this type of pattern, from outside. Another possibility is that maybe they are linked to the network structure. Maybe something about the shape of the network induces these resonances and makes these kind of like modes of the network. I'm not going to talk about vascular geometry today, but if you guys want to bring that up later, happy to discuss it. So I guess the quick answer to the thing I just said about these being linked to neuromodulatory input or vascular resonance is yes, they are. Not necessarily to one, but to both and in different ways. So one of the things we did is we did locus coeruleus stimulation. We picked one of those deep brain nuclei related to arousal, and we drove it optogenetically in rats and looked at the patterns. And we were able to see a difference in the incidence of these different patterns. It's not a huge difference. It's a fairly small one. And we think we saw it no matter what type of stimulation we use. And so we think there was an effect of the virus that we use to make this cells optogenetically sensitive to the stimulation, which is good, but we were unable to disentangle the effects of particular types of stimulation. We kind of got differences between the rats that had, were sensitive to stimulation and those that were not. And I can go into that later, but it is a link to the arousal system. We also looked at the distribution of 3 different QPPs, so 1, 2, and 3, in data from the Human Connectome project over the course of a single scan. And what we found is that QPP1 gets stronger over the course of a scan, and QPP2 kind of goes away. And it's plausible that people are getting relaxed and drowsy as they lie in the scanner, and that's why we're seeing this greater increase of QPP1. QPP1 is also tied to the global signal, and we know that gets stronger as people get drowsy. So a little bit circumstantial, but still interesting. And then when we go back to rats, sorry, my lab crosses species a lot, and so I've tried to indicate with a little rat or a person which one the data is from, just as a shortcut for you all. When we go back to rats and we do different anesthetics, we get a different distribution of QPP. So we think these are, they're sensitive to interesting differences in brain states. Okay, so that's that part, has something to do with it. The other question we had was about a resonance of the network. So I don't need to go into detail. People have done an excellent job of explaining all of these network modeling approaches. Basically, we took some more Human Connectome project data. We made a network model from the brain, parameterized it with some neural mass models. And then once we had it running, we did QPP analysis to see if these models could account for the QPPs. And what we found is that if your QPP from actual data looked like this, and I should explain how this is plotted. I said it's a movie, so this is time along this axis, and each of these along this axis is a parcel. So it's showing you what becomes active and what becomes deactivated over time. If the real data looked like that, the model data looked like this for one model or this for another model. And so you can see we're kind of getting the sort of two-phasic solution that we see in the QPP. So there's something there that maybe could come from just network resonance, but it lacks the complexity of what we see empirically. And part of that may be that the network models that we use didn't include physical adjacency as part of the model. And I think, especially when we're talking about propagation along the cortex, that's also important. You'll notice this work was done by Amrit Kashyap, who's in the room here somewhere back at the back, and he's going to talk later. So if you have questions about that, you can go to him. Okay, and just the last piece. So I said, that these patterns are kind of linked to arousal and to these large-scale processes, neuromodulation. We recently have been part of a collaboration that has shown that the same sort of cyclical activation and deactivation occurs in systems throughout the body. So that this may be some kind of signature of an autonomic signaling process that's body-wide, which I really love because it puts the brain back in the body where it really, evolved to do its work. Okay, so with all of that said, trying to get back to this multi-scale activity, we've been trying to do this a long time. We started by doing simultaneous microelectrode recording and fMRI ages ago. Other people have done this too. And this is great. It helped us to understand the resting state fMRI signal. But you've only got 2 recording locations for that one modality, right? So very little spatial information, lots of temporal information. So we started with that, but we've recently gone, oh, sorry, I have one more thing. So we saw a plot of the LFP power versus the bold signal. We looked at different frequencies. Here you can see there is a nice relationship, but it's very messy. And part of the reason we think that it's very messy is because we're only looking at the LFP from this localized place by the electrode and not at the whole brain pattern. So that was part of what encouraged us to try to find multimodal data or ways to get multimodal data that cover more of the brain. And so this is what we went to. We got excited about wide field optical imaging because wide field optical imaging lets you image neurons directly. Major failing of fMRI, right, is that we are looking at the hemodynamics instead. This lets us get the neurons directly. You can even label different types of neurons. So it was super cool. We're like, okay, we want to do it, but we want to do it with fMRI. And we were playing around with this a little bit, struggling and wondering if we could get it to work. And then Eve Lake did a beautiful demonstration of one of these setups and showed that it was possible. So that encouraged us to really put some time into it. And now we have this setup where we have a camera-based system that brings light in, illuminates our mice that have fluorescent labels and some type of neuron, and then reads out that information while we're doing MRI. So all of this goes in the scanner. And down here, this little picture in the corner, this is the tube lens that is bringing that signal out of the scanner and into the control room where we have our cameras. So this has taken a long time, but we've just gotten our first full set of data. And we're working on some things with it. I'll show you a few of the solutions that we've come up with for some of the challenges we've had. But mostly I'm going to show you that little bit of multimodal analysis that we've started to do with this data now. So because we want to do the wide field optical imaging, we need to do a craniotomy. We don't remove the skull, but we thin it. That preserves the physiology as best as possible and still gives us a very strong optical signal. I don't know if anybody's worked with fMRI in subjects or animals where you've done cranial surgery. It tends to really disrupt the image. You get these susceptibility artifacts everywhere. So we spent a lot of time figuring out ways around that. And I'm happy to talk to people if you have any interest in that. We had to register the 2 modalities. So for fMRI, we're used to just taking our 3D brain volume, taking an atlas and going, line them up, there we go, okay? But then we have this optical window that's a round port, more or less centered over the cortex, not exactly, and somehow we've got to align that with everything else. Turns out you can see the vessels really beautifully in the optical data, and so we get an MRI angiogram and use those to align, and that's how we line our optical up with the MRI with the atlas. So here are some of our first examples. We have fMRI data up here. This is just seed-based functional connectivity. So we put a seed here, see what's correlated with it. This is one scan from one animal. We've done this a lot now. I actually do have group data on this, but to emphasize the quality, I think it's important to say that this is just one scan. So we get this really nice fMRI connectivity map. And on the same atlas, we can see our raw, in this case, it's a voltage indicator image. I didn't mention, but while we do this, and I'm going to tell you why in a minute, in between our fluorescence images, we also image hemodynamics with optical intrinsic signals. So we're getting that too. And so we have a functional connectivity map from that. And then we're going to do some sort of correction I'll tell you about to get rid of that hemodynamic contribution and get pure voltage. So it works for voltage. It works for calcium indicators. Most of what I'm going to show you today is for calcium indicators. And it also works for different types of EPI sequences like not EPI, fMRI sequences like ZTE. And if you have any interest in that, I can talk to you about it later. Okay, so back to this hemodynamic contamination. The thing about the optical imaging of the calcium is that whenever we're trying to put in light to see the fluorescence that comes back out, we're also getting contributions from the hemodynamics because the hemoglobin is very absorptive, right? So if we look at our raw calcium signal here and our raw hemoglobin signal here, they're really highly correlated. You can see that they look the same. And we don't think they ought to look the same. We know if you do electrophysiology, you get the neuro response, and then you have this hemodynamic delay, right? And then you get the this response we would see with fMRI. But here we're getting it all at once, and that's because of that absorption from the changes in hemoglobin. So we have to clean that up if we want to get a clean measure of neural activity. So because we've got this interleaved acquisition system, we're able to regress out the hemodynamics from our fluorescent signal, whether it's for calcium or for voltage. And when we do that, now instead of having our calcium signal aligned at a time lag of 0 with the fMRI, we have a time lag of about 4 seconds, which is just what we would expect in an animal under anesthesia. And the same thing is true if you look at the direction of the signal. Because the hemoglobin absorbs light, you tend to get a negative response when we think we should get a positive response. And so all of this gets fixed and suggests that we're doing a good job of correcting for that. So we feel like we're actually looking now at clean calcium or voltage signals. So another thing that we can do is we can break these down into different frequency bands because, of course, optical imaging is able to go much faster than bold. And so here you'll see in the bottom row, this is the corrected voltage signal or the corrected calcium signal. We're still looking at seed-based correlation here. So the seeds are just in one of the barrel cortexes. And here we've broken this as a function of frequency. So as you go to higher and higher frequency, we start to lose bilaterality in the connectivity. Of course, with the hemodynamic signal, you actually lose that specificity much earlier because of that inherent low pass filter, just as we would expect. Okay, so now getting to some of the analysis. So for what for kind of our first approach, instead of using the whole brain MRI data that includes the subcortical structures, we have basically just taken what we can project back up to this Allen cortical atlas for a direct comparison with the optical images. And so here we have a functional connectivity matrix based on the MRI for the cortex. Here is the same thing for calcium fluorescence. And here is the same thing for that intrinsic hemodynamic signal. And this is from 10 mice under isoflurane. So we do have group data up to this point. You can see a lot of the structure is kind of preserved across modalities, which you would expect it to be. And if we look at correlation across modalities, it's about 0.5 for MRI in either of the optical techniques, about 0.8 for the fluorescence and the hemodynamics, both with optical, which seems pretty good. So we feel like we're getting the same structure. We did repeated scans in a few animals. Here's an example of one. And again, you can see that the structure is fairly well repeated on different days. So now we're at a single scan, single animal level again. And I have to keep saying this because this data is so beautiful. And that's not my doing, that's Wenju Pan's doing, because he is the one who implemented this and made it work and made it awesome. Same with the frequency dependence. I'm just going to kind of zip through this a little bit because I want to leave time for the discussion. You can make the same matrices. We start to lose specificity with the hemodynamics, of course. Okay, so one of the questions that we're really interested in is neurovascular coupling. And the reason that we're interested in this is because whenever we do something based on resting state fMRI or even task-based fMRI, we usually are assuming that the neurovascular coupling is the same across areas, across people, all the time. It's the same. And we thought, well, it's probably not the same, but how different is it really? And so these are just the first 4 animals that we did. And what I'm showing here are across this horizontal axis is 1 parcel from the Allen Brain Atlas. And the vertical atlas is the cross correlation. So we see strong positive correlation at about 4 to 6 seconds, which is what we would expect in these animals. And what you can notice is that we see that in this animal especially pretty consistently. There are some parcels that don't really seem to have as strong a correlation. I would like to know where those are, but I don't know yet. That's something I need to dig into. This animal has more variability. Here we have some areas that peak at more like 3 to 4 seconds and others that peak at more like 6 to 8 seconds. And then there's another animal that just seems to have very fast responses. I don't know if he was under very light isoflurane and about to wake up or what was going on. So there is some variability, but it's actually more consistent than I was afraid it might be. So this is something that we intend to study a little bit more. Okay, so this is where things get really dicey. And if you're at all put off by single animal results, now is a great time to go get some coffee or just disappear. I won't hold it against you. Okay, so we wanted to do at least a little bit with the dynamics, right? So those functional connectivity matrices are great, but the whole point is to start looking at dynamics. So we thought, well, what's the most simple thing that we could do? And we thought, well, maybe we should just look at modularity to start with. So we can look at modularity, on the static functional connectivity matrix. Basically, you're looking at which areas are more likely to interact with each other than with other areas. And so we did that for the different modalities. And I should apologize here again because I'm showing you data the way I like to look at data when I'm looking at it raw, not the way I usually present it. So I beg your pardon for these graphs, but this is, again, a parcel. This is the module assignment for the hemodynamics for the same animal, the module assignment for the fluorescence for the same animal, the module assignment for the MRI for the same animal. And what I'm looking at when I see this is I go, oh, well, these areas are all in the same module in MRI. Hey, they're kind of in the same module in fluorescence and kind of in hemodynamics. So just trying to get an idea of how the modular structure goes across these different modalities. If you look at the actual modularity index, it's much lower for MRI than it is for fluorescence or hemodynamics, which is probably not too surprising. I looked at frequency dependence, nothing really surprising there. Here now, this is showing for each animal along this vertical axis, and for each module in each animal, how many points were assigned to that. And all that you need to take from this is that there were a lot of modules that didn't have very many points assigned. And so for further analysis, I got rid of those. Anything that was below about 5%, I said, I'm just gonna ignore for now. I'm only gonna look at the big modules. And so I'll show you that, how that plays out in terms of figuring out how things transfer across modalities. We looked at, if you're in the same module in MRI, are you likely to be in the same module in the fluorescence or hemodynamic images? And the percentages were actually pretty good, 65 to 80 something %. So the modular structure is kind of carrying across modalities. And if you look at the modularity index up here, this is now plotted for individual rats. This is still on that time averaged matrix. Even though the MRI values and the fluorescence and hemodynamic values are very different, you can see if a rat is low on one modality, it tends to be low on all of them. So there's something that's transferring across. Okay, so I said we want to get to time varying. So the first thing we did is, okay, we're going to just chunk this time course into windows and do the same thing with modularity. We thought this is kind of a description of large scale features, and so maybe this would be a good way to start. So we took the time courses, we chunked them into 30-second segments, made functional connectivity matrices for each of those, calculated Q, the modularity index. and then just plotted that over time. And when we did that, the correlations for the Q time courses for fluorescence and hemodynamics and MRI are all close to 0. So, all right, I don't know. I don't know why that is. I don't know if it has to do with we need to tweak our modularity algorithm, but this was not the big kind of relationship that we were hoping to see for it first. So we went ahead and kind of left this for now and have gone on to other methods of looking at this relationship. So I talked about the coactivation pattern analysis before, and we have reason to believe that the same spatial patterns occur at different timings. And that comes from previous optical studies where they looked at very slow cortical fluctuations and then faster cortical fluctuations, and they were able to decompose these into the same patterns. So there were patterns that showed up in the 0.05 hertz sort of frequency range, and then the same spatial patterns occurred, but changed much faster at, say, the 3 to 4 hertz range. So there's some sort of carryover across these frequencies. So we looked at the coactivation patterns, and I'm showing a couple of what they actually look like for the cortex up here for fluorescence and for hemodynamics. You can see kind of this classical anti-correlated pair structure. You get that a lot with coactivation patterns. And so that's what it looks like. But for the further analysis, we went back to that parcellated cortical representation. And so here are all 6 coactivation patterns, and each of these lines is a parcel. And for each pattern, the intensity tells you how strong that particular parcel is in that coactivation pattern. I said there's 6. Why are there 6? Because I picked 6. I had run some analysis on the optical data trying to figure out what gave me the best silhouette index, and it was ambiguous. I said maybe up to 20, maybe 30. Wasn't sure what to do with that and how to relate that to the MRI, so I picked 6 for all the modalities for now and stuck with it. So this is clearly a non-optimal approach. But you can see we got patterns for all of these. I should point out we're doing this at the individual level for the fluorescence and the hemodynamics. I did this at the group level for the MRI data because we have so much less data and it's so noisy. Okay, so there's those patterns again. Then we looked at the correlation between different spatial patterns to see, do we see the same patterns with MRI and with fluorescence, for example? And the answer is, at least in some sense, yes. So we have high correlation between MRI cap 3 and fluorescence cap 4, for example. So we go up to here, MRI cap 3 is this, and it does indeed look like fluorescence cap 4. So we are preserving spatial information, spatial structure across these very different timescales and modalities. And then you can also go and say, okay, when I showed kind of the thought experiment part earlier, you can say, okay, well, the MRI coactivation pattern, let's say it lasts for a second. What is likely to occur during that second? And what we do see is that for a given MRI pattern, you have a higher incidence of certain fluorescence patterns. Now, these are not scaled. I realize some of the patterns occur more often, and I haven't taken that into account here or anything. But I think this is encouraging, and I think it's cool, and I'm hoping you all can help us figure out better ways to do this. So I have one last thing. This is the one I finished before I got on the plane, and was finished sticking in this morning. This is from one mouse. So what I did is we ran the QPP algorithm, and I pulled out that first spatiotemporal pattern, and I pulled out the phase. And for the phase of the MRI QPP, I plotted for each cap from the fluorescence data where it occurred. And so when you see kind of a bulge like this, that means that cap 1 was more likely to occur around this phase and much less likely to occur around this phase. And so I think this is encouraging to me that we see different caps occurring at different phases, some maybe even with a bimodal distribution. I really want to look into that. It looks like maybe that clusters around the switching area between the positive and negative kind of portions of these spatiotemporal dynamics. But all that said, this is one mouse. So, take it with a grain of salt. But that is really the last bit. I just wanted to kind of wrap up really quickly. We think brain states are a good way to kind of look at these things across scales and brain trajectories, especially if we can get to that. We're very excited about this wide field optical and fMRI approach because it finally gives us some really rich multimodal data with different scales that we can work with. We're excited about targeting the fluorescence indicators to other things, inhibitory neurons, astrocytes, and seeing how they play into this. But the number one for everybody here is that there's a lot to do to understand how we put these things together experimentally. And I'm hoping you all can help us with that. We have code and data available. And I want to make sure I acknowledge the lab, and especially Wenju Pan, who's been a research scientist working with us for a long time and is really the person I have to give all the credit to for getting this optical and MRI system up and working. Okay, and we're the Pirate Lab, so random last slide. So thank you and happy to take questions.